Welcome back to MLE Masters, your daily dose of high-yield USMLE content. In today's lecture, we're diving deep into one of the most commonly tested topics in genetics, meiosis errors and chromosomal abnormalities. Let's understand how even the slightest error in meiosis can lead to significant genetic consequences, and syndromes you'll encounter both on exams and in clinical practice. Let's start by understanding what exactly meiosis errors are. During gamete formation, chromosomes are supposed to segregate equally between daughter cells. But when this process fails, it's called nondisjunction. This can happen during Meiosis I, when homologous chromosomes fail to separate, or in Meiosis II, when sister chromatids don't divide properly. The result is aneuploidy, an abnormal number of chromosomes, which is the basis of many genetic disorders. So what happens when nondisjunction occurs? We can get a trisomy, which means there's an extra chromosome, or a monosomy, where a chromosome is missing. While most monosomies are incompatible with life, some trisomies like Down syndrome, Edwards, and Patau can result in live births. Each of these carries its own clinical significance and unique features. Down syndrome, also called trisomy 21, is the most common autosomal trisomy that results in a viable birth. It's characterized by distinct facial features like flat facies and epicanthal folds, intellectual disability, and a single palmar crease. You'll also see associated congenital heart defects, particularly atrioventricular septal defects. Leukemia and early-onset Alzheimer's are important associations too. Most cases occur due to maternal nondisjunction during Meiosis I. Next is Edwards syndrome, or trisomy 18. These patients present with severe intellectual disability, rocker bottom feet, micrognathia, low-set ears, and clenched fists with overlapping fingers. Congenital heart defects are common, and unfortunately, survival beyond the first year is rare. It's a high-yield condition for test questions. Let's move to Patau syndrome, or trisomy 13. This disorder presents with microcephaly, hollow prosencephaly, cleft lip or palate, and polydactyly. Congenital heart defects are also seen. It's a severe disorder with poor prognosis, and most affected infants don't survive beyond early neonatal life. Now let's discuss Turner syndrome, the only viable monosomy in humans with a karyotype of 45X. These individuals are phenotypically female and present with short stature, webbed neck, shield chest, and streak ovaries, leading to infertility. Cardiac abnormalities like coarctation of the aorta and bicuspid aortic valve are common. Despite these features, intelligence is usually normal, and many cases are diagnosed at puberty. Klinefelter syndrome results from an extra X chromosome in males, a 47XXY karyotype. These males are typically tall with small testes, gynecomastia, and infertility due to low testosterone levels. FSH and LH levels are elevated due to gonadal failure. There's also a mild risk for breast cancer and learning difficulties. It's another classic step 1 condition. Beyond Turner and Klinefelter, there are other sex chromosome abnormalities. 47XYY males are often tall and may have mild behavioral issues, though they're usually fertile and asymptomatic. 47XXX females often go undiagnosed. They may have normal fertility and only mild learning issues. These are often incidental findings during genetic testing. Now let's talk about Robertsonian translocations, another important genetic mechanism. It's a fusion between two acrocentric chromosomes like 13, 14, 15, 21, or 22. While balanced carriers are usually unaffected, they can pass on unbalanced chromosomes, leading to syndromes such as translocation Down syndrome. In such cases, total chromosome number may be 45, but with all the necessary genetic material. Mosaicism refers to the presence of two or more genetically different cell lines in one individual. Somatic mosaicism affects body tissues, while germline mosaicism involves only reproductive cells. This explains why some individuals have variable presentations and why certain genetic disorders recur in siblings even when parents are normal. One of the most important risk factors for nondisjunction is advanced maternal age, especially affecting Meiosis I. This is why the incidence of Down syndrome increases significantly after the age of 35. It's a key point often tested in both clinical scenarios and epidemiology-style questions. Prenatal screening plays a crucial role in early detection. Thanks for joining us at MLE Masters. If you found this video helpful, don't forget to like, share, and subscribe to stay updated with more high-yield medical content. Drop your doubts in the comments, and as always, keep learning, keep mastering.